This is the lecture for Chapter 2, Basic Chemistry. Before you can study biology, you must have a good understanding of basic chemistry. And chemistry is defined as the composition and structure of substances. If you remember in the previous lecture, I talked about how one of the purposes of science is to organize information in a logical order. And one level of organization is going from the very smallest, the atomic level, all the way up to the biosphere. And in this, I mentioned that at this lower level is the molecule and the atom, and that is covered by chemistry. And of course, biology goes from the cellular level on up to the biosphere. So in order to understand the cells, it's very important to understand the different substances that compose the cells. On a more specific level, when we're looking at the level of organization that is covered by chemistry, it begins with the subatomic particles. When you bring those subatomic particles together, you create an atom. Several atoms that are brought together and bonded together create molecules. And in terms of biology, molecules, very large molecules called macromolecules, are the essential building blocks of the cell. So in order to understand the cell, you need to really understand the structure and function of the different types of macromolecules. As I mentioned in the previous slide, chemistry is the study of the composition and structure of substances. Substances, you can also refer to those as matter. And the basic definition of matter is anything that takes up space and has mass. And mass is similar to weight, just mass is a constant and weight takes into account gravity. So for instance, you on Earth have a different weight than you would on the moon because the Earth and the moon have different forces of gravity, but your mass is consistent. So going through this, mass is basically very similar to weight, but again, taking into account gravity. And mass is measured in grams or kilograms. And in science, we always use metric. But when you're talking about mass, you're talking about grams or kilograms. Space, that would be volume. And so volume is usually measured in liters or meters. Now, matter could be a liquid. So liquids, they have mass and they take up space. They have a volume. It could also be anything that is a solid. So again, a solid has mass and takes up volume, and it could also be a gas. And a gas also has a mass and takes up a volume. Another important term in chemistry is the term element or elements. And elements are defined as the simplest type of matter. And that means they cannot be broken down into simpler substances by chemical means. So it's like the smallest particle of matter that has that matter's unique properties, like the specific density, the solubility, its ability to dissolve, its melting point, and its reactivity. So that is the definition of an element. It's the simplest type of matter. It cannot be broken down by chemical means. And the different elements that we have discovered on Earth, 94 are naturally occurring. The rest have been synthesized by scientists, but we have organized them in the periodic table of the elements. So this is the periodic table of the elements, which I'm sure you have seen. And only 25 of these are essential for life. It is actually very interesting when you look at the elements that are used to build living things. And the interesting part is that these 4 elements, oxygen, carbon, hydrogen, and nitrogen, make up 96% of the human body weight. So 96% of your body by weight is composed of only 4 different types of elements. The rest of your body, 4%, about 4% of your body weight is made up of these other elements. Things like calcium, phosphorus, potassium, sulfur, sodium chloride, and magnesium. So that makes up about 4% of your body weight. And you do have other elements, a few other elements that make up a very small percentage of your body weight, and we refer to them as trace elements because you need them you do need them for your cells, your body to function, but you need them in very, very small amounts, trace amounts. Another important term in chemistry is the term atom or atoms. And often atom is used interchangeably with element, but they don't mean exactly the same thing. So an atom is defined as the smallest part of an element that has the same properties, and so it has the same name. So it's the smallest part of the element that behaves as that element. And the term atom actually is Greek, and it means uncut or indivisible. So you can't divide it down into any smaller particles and still have that same reactivity or those same characteristics. And again, this is the periodic table of the elements, and it gives you information on the atoms of those elements. So with this, you have the rows that go across, and those rows are called periods. So that is the periodic table of the elements. So you have the first period, second period, third period, that's going across. And then the columns are going up and down, and the columns are called groups. So this is a group, and groups of atoms or elements have similar characteristics. So this group going down here, the column here, those are the noble gases. So helium, neon, argon, they have very similar characteristics. Also compared to the group here. So that group, the hydrogen, lithium, sodium, potassium, they have similar characteristics also. And for this is color-coded with elements of greatest biological reference. So the ones in red are elements that are very common in living things. And the ones in yellow are ions that are important for living things. So as you can see for biology, most of the important elements, most of the important atoms that we'll be talking about occur in the first 3 periods for the most part, and a little bit into the 4th period. So even though atoms are described as the smallest part of an element that has the same properties, atoms themselves can be subdivided into smaller particles, and those are called the subatomic particles. And scientists over the many years have found many different types of subatomic particles. And some of them I'm sure you have heard of: proton, neutron, and electron. Also you may have heard of photons, and maybe even the quarks and bosons maybe. And there are all sorts of other ones, such as lepton, graviton, neutrinos, and dark matterite. But there are many different types of subatomic particles. In this class, we're just going through basic chemistry, so we are only going to focus on the basic subatomic particles. And so that would be the neutron, the proton, and the electron. So it's going to be important for you to understand how these come together to form an atom. So again, just focusing on these 3 types of subatomic particles, you should understand how they come together to form basic atomic structure. So basic atomic structure, you have a nucleus, and in the nucleus you have the protons, which have the positive charge, and you have neutrons, which are neutral. They don't have a charge. Then orbiting around the nucleus are the electrons. So the electrons are orbiting around the nucleus. And really, the electrons are spinning so fast around the nucleus that they move a little bit in their space. So we don't always know exactly where they are, but the orbitals that we draw, that is a probability of where we find the electrons about 90% of the time. So this is an animation of a drawing of an atom. So here is the nucleus with the protons and neutrons, and then you have all these electrons orbiting around the nucleus. Now this is a little misleading because in reality, atoms have a lot of space. So I think in your book it talks about a nucleus. A nucleus would be about the size of a marble. If you put a marble in a football stadium, the nucleus, even a large one with lots of subatomic particles, protons and neutrons, would be about the size of a marble. And then the electrons that are spinning around, that would be the outer part of the football field. So the point of this is there's a lot of empty space between the nucleus and the electrons. They're not as close as they are in this animation or in any of the diagrams that we use. So you'll want to keep that in mind. The next thing I want to do is go through the three subatomic particles, the protons, neutrons, and electrons, and talk about their importance to the atom. So the first subatomic particle are the protons, and protons are very important for atoms because they determine the element that the atom belongs to. So when you look at the periodic table of the elements, you notice that every element has an atomic number. And the atomic number is the number of protons that are found in the atoms of that element. So in this example, the atomic number 8 means that every atom that has 8 protons will be an atom of oxygen, and it will have the characteristics of oxygen. So if you look at the periodic table of the elements, you will see the atomic number at the top of each square, starting with 1, and as you go across the period, you have 2 here, and then you go down to the lower period, you have 3, 4, 5, 6, 7, 8, 9, 10, and so on. So again, the atomic number is telling you the number of protons that is in the atom. So every atom that has 10 protons must be an atom of neon, and so on. The second type of subatomic particle that I would like to discuss are the neutrons. And neutrons are very important because they have an effect on the atomic mass of the atom. When looking at the information that is given in the periodic table of the elements for each of the elements, you always have your atomic number, which remember is the number of protons, and that determines what element that those atoms belong to. And often you have the notation, which is usually 1 or 2 letters, that indicates that element. So for instance, any atom that has 6 protons is going to be an atom of carbon, and those atoms are designated by the letter C, capital C. Often at the bottom of these little boxes of information, you have a number here, and this is the atomic mass. So that is the atomic mass, basically the mass of the atoms. And often what you see is that the atomic mass is 2 times the atomic number, close to 2 times the atomic number. And that is because protons have a mass of 1 AMU, which is atomic mass unit. It used to be known as a Dalton. So protons, their mass, sort of their weight. Remember, weight is mass with gravity taken into account. So protons have a mass of 1 AMU, and neutrons have a mass of 1 AMU. And electrons have a much smaller mass. It's about 1/1000th of an AMU. So electrons don't really count when you are counting up the mass of an atom. So ideally, an ideal atom will have the same number of protons as it does the same number of neutrons and the same number of electrons. So in one atom of carbon, you would have 6 protons, 1, 2, 3, 4, 5, 6. So that designates this as an atom of carbon. And then if this is an ideal atom, you'll have 6 neutrons. So you have your 6 neutrons. So together you have 12 subatomic particles in the nucleus, and because the protons weigh 1 AMU, the neutrons weigh 1 AMU, 12 AMUs, you will get an atomic mass of roughly about 12. And also, as I mentioned before, you should in an ideal atom have the same number of electrons. So you'd have 1, 2, 3, 4, 5, 6 electrons orbiting the nucleus. So the role of the neutrons in an atom is to have an effect on the mass, the atomic mass of that atom. I mentioned that an ideal atom would have the same number of protons and the same number of neutrons and the same number of electrons, but often in nature things are not ideal. So sometimes we will have an atom that has a number of protons that does not equal the number of neutrons. And when this happens, the atom is considered an isotope. So for instance, this happens with carbon quite a lot. An ideal atom of carbon would be this. This is an ideal atom of carbon. And if you look in the nucleus, the protons are blue. You will see 6 blue protons. So that means it is a carbon atom. And you would have 6 neutrons, which means it has an atomic mass of 12, which is normal. And of course, it has its 6 electrons. So this would be an ideal atom of carbon, and it is very stable. So its structure is very stable. But sometimes in nature, you have some carbon atoms that have an extra neutron. So this is a case where the number of protons does not equal the number of neutrons. So if you look at this, this does have 6 protons, so those blue protons, which means it is an atom of carbon. It's an atom of carbon. But if you count up, it actually has 7 neutrons. So that is going to change the weight. So now you have the 6 protons plus the 7 neutrons, and now the atomic mass is 13. So this is carbon 13. So this is a carbon atom because it has only 6 protons, but it has a heavier atomic mass because it has an extra neutron. And carbon 13 is stable also. Also in nature, we have carbon 14. And carbon 14, if you count up the atomic particles, it has 6 protons, which makes it a carbon atom, but now it has 8 neutrons. So it has 8 neutrons, so 6 plus 8 is 14, so it has an atomic mass of about 14. And when you have a carbon 14 atom, it is unstable. So now the nucleus isn't very stable, considering the type of electrons that you have, which you have 6 electrons, and we consider this unstable atom radioactive, and it is an isotope. There are many elements that have different isotopes. And remember, an isotope is an atom that has an unequal number of protons and neutrons. So many atoms have different isotope forms, different isotopes. And if those isotopes are unstable, so if they're unstable, then those nuclei are going to decay. And when we say decay, what that means is that atom will release a subatomic particle. Could be a neutron, could be a proton, could be an electron, but it's going to release a subatomic particle to try to become stable. And there are different types of subatomic particles or different types of radiation that can be produced by radioactive isotopes. There's alpha radiation, beta radiation, and gamma radiation. So again, these are different particles that are released by a radioactive and unstable isotope to try to become stable. Now, in terms of living things, we are very concerned about gamma radiation because gamma radiation is the type of radiation that can damage living cells. As an unstable isotope decays, it's going to release radiation, and radiation can damage cells in 2 different ways. You can have direct radiation or direct ionization where the radiation directly damages the DNA. And when DNA is damaged, it can lead to mutations, which can either kill the cell or in a multicellular organism, it can cause cancer. There's also indirect ionization or indirect radiation where water is ionized, and then that water can cause, that ionized water can cause damage to the DNA. So that is a big concern with living things, that radiation can damage the DNA, which can lead to the death of the cell or cancer in a multicellular organism. But there are some radioactive isotopes that are helpful, especially in diagnostics in medicine. So different diagnostic tests will use different types of isotopes in their tests to identify different conditions. So not all radiation is as damaging as gamma radiation causing cell death or cancer. So some radiation is not as damaging to the DNA. The last subatomic particle I want to go through are the electrons, and the electrons are very different from the protons and neutrons. First of all, the location. Electrons are found outside of the nucleus. So those protons and neutrons are in the nucleus. They compose the nucleus. The electrons are found outside the nucleus. And whereas the nucleus doesn't have a lot of motion, the electrons are spinning very fast around the nucleus. And it's very difficult to anticipate the actual location of an electron because they're moving so quickly and a little erratically, but most electrons can be localized to a particular orbit or a particular shell. And they can be found in this particular orbit about 90% of the time. Not 100% of the time, but 90% of the time. The other thing about the electrons, which you probably know, is that they have a negative charge. So they have a negative charge. Protons have a positive charge. Neutrons have no charge. And the other thing about the electrons is they have a very small mass. It's about 1/1840th of an AMU, or a Dalton. So they're very small, electrically charged, and zooming around the nucleus, so orbiting around the nucleus. One of the important functions or correlations regarding electrons is in terms of energy. So the location of the electron from the nucleus relates to how much energy that electron has. So looking at the atomic structure, so here, down here, we have the atomic nucleus. The atomic nucleus has the protons and neutrons in it. Then outside, surrounding that atomic nucleus are the electrons. And the electrons can come in 3 different shells. And shells are like orbitals. So you have the first shell, which is very close to the nucleus. You have a second shell, which is further away from the nucleus. And you have the third shell, which is further away. Now, the location, which shell the electron is in, relates to its energy. So the first shell, the closest to the nucleus, the lowest energy, the lower the energy the electron has. As it moves away from the nucleus, the energy potential that the electron has increases. So electrons that are found in the outermost shell have the highest potential energy. Electrons on the innermost shell have the lowest potential energy. So what that means is for an electron to move from the first shell to the outer shell, it needs to absorb energy. So there needs to be an energy input to get it enough energy to boost it up to that highest level. And conversely, when an electron goes from a high level down to a low level, it's going to lose energy. So as that electron loses energy, it's going to fall to a lower level. And this understanding how electrons and their location from the nucleus and how it relates to energy is going to be very important when we talk about photosynthesis, when organisms harvest the energy of the sun and transform that energy into chemical energy. So this concept is going to come back when we talk about photosynthesis. When talking about electrons, it's very important to note the valence electrons. And the valence electrons are the electrons that are in the outermost shell. And these are the electrons that are going to have the most energy, and they're also going to be involved in chemical bonding. So to understand where the valence electrons are and how reactive they would be, you need to have a basic understanding of the different shells. Now, for biology, for people interested in biology, we're only interested in the first 3 shells that are filled by the electrons. So in period 1, so this would be the first row in the periodic table of the elements, that only involves one shell, so that would be the innermost shell. Period 2, which is the second row, all of those elements or atoms of those elements have two shells. So here's the inner shell and here's the outer shell. And period 3, which is the third row, has 3 shells. So we're mainly going to focus on those. Now in each shell, for a shell to be completely filled with electrons, you would have a pair of electrons. So electrons like to kind of go in pairs. Think of it that way. So in this group, so that group, that column, these are all completely filled shells. All right, so in the first shell, the maximum number of electrons that can go there is 2. So if an atom has 2 electrons in its first shell, that first shell is completely filled, and then that atom is stable. All right, period 2, the second shell, the maximum number of electrons you can have are 8. So you have 4 pairs of electrons. So that means that that shell is completely filled and it is very stable. Now when adding electrons to a shell, so when electrons are added to a shell, the way they're added is you add 1 electron of each pair first. So if you have 1 electron in the second shell, it would be there. If you have 4, you wouldn't have 2 pairs. Instead, you would have 1 of each potential pair. So 1 of each potential pair. And then as you add more electrons, then you start filling out the pairs. So you start filling out the pairs. And then, of course, you add 1 more to this pair. That would create a stable shell. And the third shell is very similar to this. It also holds 8, a maximum of 8 electrons. And again, you're going to start filling them up. First 1 electron for each pair, and then you start filling in the pairs. So ultimately, you would have 4 pairs of electrons in the outermost shell. The importance of understanding how many valence electrons an atom has, it will give you information on the bonding, the type of bonding that that atom will be involved in. And when I say bonding, I mean forming a molecule with another atom. And it's important to remember that atoms are more stable if they have a filled valence electron shell. So if their outermost shell has the maximum number of electrons. So if you're looking at an atom like hydrogen, which has an atomic number of 1, so it has 1 proton in the nucleus, and ideally it has 1 electron in its first shell. You look at this and you see that this first shell is not filled because it can have a maximum of 2 electrons. So that would tell you that this hydrogen atom is going to be reactive because it's going to want to fill that missing spot with another electron. And to do that, it can bind or it can bond to another atom. Another important type of atom are carbon atoms, and carbon atoms have 2 shells. Their innermost shell has a pair of electrons. They're not drawn together, but it has 2 electrons in the inner shell, so the inner shell is complete. But in the second shell, it has 4 electrons, so 1 of each pair. So for a carbon atom to be complete, it would need 4 more electrons to help fill that outer shell. Nitrogen is similar to carbon, very close, but it has an inner shell which is complete with a pair of electrons there, but in its outer shell it has 5 electrons. 1, 2, 3, 4, 5. So if you were going to draw this a different way, just the outermost shell, it had 5. It has 1, 2, 3, 4, and then it started to fill this pair of electrons, so that's set, but now it's going to look for 3 more electrons to complete those pairs. And oxygen, another important atom for living things. Oxygen has a completed inner shell, so that's set, and then it has 6 electrons in the outer shell, 1, 2, 3, 4, 5, 6. So if you were going to fill this outer shell out, that's O for oxygen. That's the nucleus. So 6, 1 of each pair first, 1, 2, 3, 4, and then filling out the pairs. 5, 6. So it's going to be looking for 2 more electrons to complete its outer shell. Sometimes it happens in nature where an atom has a different number of protons and electrons. So in this case, some atoms, the number of protons does not equal the number of electrons. And when this happens, the atom is going to have an overall charge, and then it is called an ion. So in normal situations, when the number of protons equals the number of electrons, that means the number of positive charges is equal to the number of negative charges, which means overall the atom has a neutral charge. But if you change that, if you have unequal numbers of protons and electrons, then the atom will have a charge. So one example of this is sodium. Sodium, its atomic structure, it has 3 shells of electrons, and in the outermost shell, in the third shell, it has only 1 electron. So in order to be stabilized, it would need to find 7 more electrons to fill its outermost shell. So an easier way to get to stability, instead of finding 7 electrons, is to give this electron away. So what happens often with the sodium atoms is that they give away their electron, they release their electron. So now they are left with only 2 shells, but those 2 shells are completely filled with the maximum number of electrons for those shells. So now this has stabilized the sodium atom. But in giving away an electron, it has removed a negative charge, and now the overall atom has a +1 charge because now there's 1 less electron compared to the protons. So this often happens with atoms like sodium. So atoms like sodium, which have only 1 electron in their outermost shell, they're much more likely to give up that electron. And then often what happens to that electron, electrons aren't usually floating around free in the environment. What happens is that electron is then taken up by a different atom that just needs 1 electron to complete its shell. So example, chlorine. Chlorine in its third shell, it has 7 electrons normally. 1, 2, 3, 4, 5, 6, 7. So all it needs is 1 more to have a completed valence electron shell. And since sodium is ready to give up its electron, often chlorine will take that electron, and then they both have completed valence electron shells. And in this case, the chlorine is going to have a negative charge, a net negative charge, a negative charge because it has gained an extra electron. So just to summarize what I've gone through with the subatomic particles, the proton, neutrons, and electrons, I want to put together some basic information in a table so that you can compare and contrast the 3 subatomic particles. So the first thing is the charge. Protons have a positive charge, neutrons have a neutral charge, and electrons have a negative charge. And in terms of intensity, the level of the positive charge of the proton is equal to the power of the electron's negative charge. So if you have equal numbers of protons and electrons, then the overall atom will have a neutral charge. In terms of mass, protons are 1 AMU, atomic mass unit. Neutrons have the same basic mass, 1 AMU, and electrons are much, much smaller. So they have 1/1840th of an AMU. So for all intents and purposes, when you are calculating the atomic mass, you only need to know the number of protons and neutrons. You add those numbers together, and that will give you the mass. Electrons have such a low mass that you don't really need to take them into consideration. In terms of location in the atom, protons are in the nucleus, neutrons are also in the nucleus, and electrons are in the orbitals or the shells. Orbitals or shells, however you want to refer to them. So the protons and neutrons are together in the nucleus. The electrons are orbiting in shells around the nucleus. Now, the last thing I just want to go through is the role or the purpose or what they do for the atom. I don't know exactly how to refer to it, but the number of protons will determine the element. So remember, the number of protons is going to determine what element the atom belongs to. So if it has one proton, it has to be a hydrogen atom. It has no other choice. And so it's going to behave with all the characteristics of hydrogen. Neutrons. Looking at the number of neutrons that an atom has, this is going to determine if the atom is an isotope or not. So remember that the ideal atom has the same number of protons, neutrons, and electrons. So if you look at the number of neutrons, that's going to change the mass, which will indicate if that atom is an isotope of a particular element. Then electrons. Electrons have sort of a lot of functions or a lot of indicators in an atom. So the first thing with the electrons is that if you have unequal numbers, if you have a different number of protons and electrons, then you are going to have an ion. Then you know the atom is an ion. Actually, and also understanding where the electrons are in relation to the nucleus will give you information on their level of energy. And then the last important thing about the electrons is understanding how many valence electrons are in the outermost shell. This will give you information on how they're going to form chemical bonds, which will be very important. In chemistry, if you look at the increasing level of organization, you start with subatomic particles like the electrons, protons, and neutrons, and those can come together to form the next higher level of organization, which would be the atom. And then when you have one or more atoms bonded together, that is when you get a molecule. So that's the next higher level of organization. So just as an example, atoms of an element, these are individual atoms of an element, and some elements consist of just individual atoms. Other elements, because of the configuration of the valence electrons, their molecules are two atoms of the same element bound together. So this would be like hydrogen, because I mentioned hydrogen has only one valence electron, so it really wants to complete its shell. So often with hydrogen as the element, it has two atoms bound together so that they have a stable configuration. So that's a molecule, two atoms bound together. But when you have two or more atoms, different atoms bound together, then it's a compound. So this is an example just by color. You have three atoms, green atoms of green element, bound to one purple atom, so purple element. So now this is a molecule, but it's also called a compound because it has at least two different types of atoms. Later in the semester, when we talk about different biochemical processes that occur in cells, such as photosynthesis and cellular respiration, we're going to be referring to different types of molecules by their chemical formulas. So the chemical formula is a way to represent what atoms are found in the molecule. And so H2O, water, I'm sure you are familiar with. Water is composed of two atoms of hydrogen, one atom of oxygen. So a molecule of water, which is a compound because it's composed of two different types of atoms, it is written out as H2O. So two atoms of hydrogen bound to one atom of oxygen. And here are some other molecular formulas. Hydrogen gas, which I mentioned before. Hydrogen, because each hydrogen atom has only one valence electron, they often bind together to stabilize themselves, and so these are different ways to represent the molecules of hydrogen, the element hydrogen. And then oxygen also binds together to share two pairs of electrons, so often the element is written this way, and water, H2O, and methane is one carbon, which is bound to four hydrogen atoms. So it's a good idea to be able to read the molecular formula so that you can follow how many atoms and what type of atoms are in a molecule. In order for atoms to bind together to form molecules, they need to form chemical bonds. And there are several different types of chemical bonds that I want to discuss, and the first one are the ionic bonds. Ionic bonds are formed between ions. And remember, an ion is a charged atom. So in the example I gave before, sodium has only one electron in its third shell. So in order to stabilize, to have filled valence electron shells, it's much easier for it to give away that electron rather than trying to find 7 more electrons to fill its shell. Chloride, on the other hand, or chlorine, on the other hand, it has 7 electrons in its outermost shell, so it just needs 1 more to complete its shell. So it's more likely to take that electron, add it to its third shell, and then have a completed shell. And in doing so, in doing this transfer of an electron, the sodium ion is giving away an electron, so overall the atom is going to have a positive charge. When it has a positive charge, it's called a cation. The chloride is going to receive that electron, so it's gaining electrons, so overall the atom is going to have a negative charge, and that we call an anion. And this is just an animation showing the same thing, where the sodium is giving away its electron to a fluorine in this case, which has the same situation as the chlorine, and creating a cation and an anion. So ionic bonds are the interaction between 2 oppositely charged ions, between an anion and a cation. And the bonds that hold them together are basically like magnetic bonds. So if you have something that has a positive charge, it's going to be attracted to something that has a negative charge. So every positive ion, cation, is going to attract negative ions around it. And every negative ion is going to attract positive ions around it. So what you end up getting are these lattice structures. So if the green ones are the cations, they have a positive charge, and they're going to be surrounded by negative or anions. And that creates what we call a lattice structure or a crystal structure. And ionic bonds are very, very strong, almost impossible to break, and they are strong enough that living cells, the enzymes in the living cells, cannot break ionic bonds. So ionic bonds are not found in living things. So in a living cell, molecules you find in a cell, they do not have ionic bonds. A second type of bond that an atom can form with another atom is called a covalent bond. And the covalent bond is where the atoms are sharing, pairs of electrons. So that's a covalent bond. So co together, valence electrons. So now instead of giving away or accepting an electron, now the 2 nuclei are sharing pairs of electrons. And one example is hydrogen gas. So remember, each hydrogen atom has only 1 electron, and each one has only 1 electron, and they want a completed valence shell. So because it's in the first shell, they just need a second electron. So what they do is they share this pair of electrons. So that pair of electrons is going to be orbiting both nuclei. So both of them will orbit each nuclei. So they're sharing pairs of electrons. And when they do this, this is bringing the nuclei physically close together. With ionic bonding, the nuclei are pretty far apart because they're held together by electromagnetic interaction, by charge interaction. But with covalent bonds, since they're sharing pairs of electrons, the nuclei are physically close together. So this is an example of a single covalent bond because they're sharing 1 pair of electrons, and we designate this with 1 line between the 2 atoms. Sometimes you will have atoms sharing 2 pairs of electrons. So now you have, with oxygen, 2 pairs of electrons that are being shared, and that's called a double bond. So now instead of 1 pair of electrons orbiting the nuclei, you now have 2 pairs of electrons orbiting the nuclei. And the last type of covalent bond you can have is a triple covalent bond. So it's represented by 3 lines because you're sharing 3 pairs of electrons. And the 3 pairs of electrons, again, just like the other ones, are orbiting both nuclei. Covalent bonds are strong bonds. So the molecules that are made with covalent bonds are stable molecules, but it is possible for living cells to be able to break covalent bonds. So living cells can form covalent bonds and break covalent bonds, unlike the ionic bonds where living cells are unable to break ionic bonds. There are 2 subtypes of covalent bonds, and the first subtype of covalent bonds are the nonpolar covalent bonds. Nonpolar means that the electrons are shared equally between the 2 nuclei, and this is because you have the same charge on both nuclei. So for instance, for hydrogen, both nuclei just have 1 proton, so they're balanced, so the electrons are equally shared. So they orbit each of the nuclei equally, and so overall the molecule is uncharged. So it doesn't have a charge, it doesn't have a positive area or a negative area. So that is nonpolar covalent bonds. The other type of covalent bond are the polar covalent bonds. And polar covalent bonds means that there is unequal sharing of the electrons. And a typical example of this is water. So here's your water molecule, 1 oxygen bound to 2 hydrogens. Now the reason there is unequal sharing of the electrons, this indicates the sharing of a pair of electrons. The reason it's unequally shared is because of the number of protons in the nuclei. Hydrogen has an atomic number of 1, so there's only 1 proton in each of the nuclei of the hydrogen atoms. Oxygen has an atomic number of 8. So it has an atomic number of 8, so it has 8 protons in the nucleus. So it has 8 protons. 8 protons have a positive charge, whereas the hydrogens have only 1 positive charge in their nuclei. So when these pairs of electrons are being shared, this charge of 8 protons is going to draw the electrons over towards the oxygen for a longer time than the 1 positive charge with the hydrogen. So what you end up with is unequal sharing. So when the electrons are orbiting, they spend more time around the oxygen because of this higher positive charge than they do around the hydrogen. And this happens on the other side too. So on the other side, the pairs of electrons are going to spend more time around the oxygen, less time around the hydrogen's nuclei because of the difference in charges of the nuclei. And we call that electronegativity. So the oxygen has higher electronegativity. It's drawing the electrons to it more often. So what happens is because the electrons are spending more time orbiting around the oxygen's nuclei, you tend to have a negative charge on that side of the molecule. And because the electrons are spending more time on this side of the molecule, they're spending less time on this side of the molecule, so this tends to have a positive charge. And that's what the sigma means. Sigma negative means that this side of the molecule, the oxygen side of the water molecule, tends to have a negative charge, whereas the hydrogen side of the water molecule tends to have a positive charge. And not only is this unequal sharing of electrons, but it also creates, it's not a symmetrical molecule. So actually the 2 hydrogens aren't straight across, they're at an angle here because all the electrons are spending all this time up there. And with this polar covalent bonds, this is often found in molecules like water that contain an oxygen, which is highly electronegative, and also in molecules that have nitrogen in them, like ammonia, because nitrogen is also highly electronegative. For the rest of this lecture, I'm going to talk about a very special molecule, H2O, which is water. I'm going to talk about this because H2O, water, has very unusual characteristics, which makes it a very important molecule for living things. I've talked about the structure of water a couple of different times, but just to review it, water, the chemical formula is H2O, so you have 2 hydrogens bound to an oxygen. And in terms of the atomic structure of the hydrogens, each hydrogen has one proton and it has one electron orbiting around that one proton. The oxygen, on the other hand, has 8 protons and 8 neutrons. And then it has 8 electrons orbiting around that nuclei. And in order to complete the valence shells for all the atoms, the hydrogen has one covalent bond with the oxygen, and the other hydrogen has a covalent bond with the oxygen. And that gives all the atoms a completed valence electron shell. And remember that this forms a polar covalent bond because this nucleus has such a high positive charge. The electrons tend to spend more time around the oxygen's nuclei and less time around the hydrogen's nuclei. So the hydrogens tend to have a positive charge, whereas the oxygen tends to be slightly negative. And being a polar covalent molecule, this gives water a lot of unusual characteristics. The main characteristic that makes water so unusual is its ability to form hydrogen bonds. And hydrogen bonds are bonds between molecules. So for this hydrogen bond, a hydrogen bond occurs between polar molecules. So in this case, this is a water molecule. And remember, the oxygen has a partial negative charge and the hydrogens have partial positive charges. So what that means is in liquid water, or in water, a sample of water, one molecule, the partial negative charge of one molecule will be attracted to a partial positive charge on another molecule. So all water molecules are going to be hydrogen bound to other water molecules. Now, the interesting thing about hydrogen bonds is that they're very, very weak. So that's why we draw them with a dotted line, because they're very weak. They are continually being broken and reformed. And this kind of shows you a little bit. Here are 3 water molecules that are hydrogen bound together. So they're a little bit of flexibility. They're kind of flexing a little bit, but they're held together. Now, the thing about hydrogen bonds is each individual bond is extremely weak, and actually this animation should show them breaking and reforming. But when you get a lot of hydrogen bonds together, you get a very strong force. And an analogy I have used before in class is Velcro. So in Velcro, you know you have 2 strips of Velcro. One strip has little hooks on it. The other strip has little loops on it. Now, every individual little hook interacting with a little loop, that would be a very weak interaction. It wouldn't hold together very much. But when you have hundreds, thousands of hooks interacting with hundreds, thousands of loops, now you get a strong interaction. And they have industrial Velcro that is often used in safety harnesses. So this is industrial Velcro. It's used for safety harnesses. So if this man falls down, the Velcro will hold, his harness will hold, and he won't fall to the ground. And that is because the numbers, you have lots of loops interacting with lots of hooks, you get a very, very strong force. And that is true with hydrogen bonds. Each individual bond is very weak, but when you get many of them together, hundreds of thousands, billions of them together, you're going to get an incredibly strong force. And yet, those molecules still have flexibility to move. There are several different characteristics that make water really important to living things, and the first is water's ability to have a high heat capacity. And what that basically means is that water can absorb a high amount of energy before it changes temperature. And the reverse is true too. So water can release energy, a lot of energy, before changing temperature. And that is all due to this hydrogen bonding. Because these bonds are individually weak, but together a very strong force, it takes a lot of energy to completely break the bond and release the molecule as steam. So that's why when you put a pot of water on top of the stove to boil, you put the water on the stove and then you turn up the gas flame really high, and then it takes a long time 10 minutes or so under that high temperature before you actually start getting significant amounts of water leaving as steam. And that's because these hydrogen bonds can absorb a high amount of energy before they completely break and release the molecules. Having a high heat capacity means that water is a good insulator. And not only does it insulate living things, it helps us to maintain our homeostasis with our temperature, but it also moderates temperature in the environment. So you may be aware that areas that live near large bodies of water, like the Pacific Ocean or even Lake Michigan, they have more mild temperatures than areas that are not surrounded by a large body of water. And that's because as the sun releases more energy on the Earth, the water is going to absorb a lot of that energy and maintain a more mild temperature. Whereas if you don't have that water, then that energy from the sun is going to increase the temperature. So water has this ability to insulate or moderate temperature changes. Another unusual characteristic of water is that solid water, ice, is less dense than liquid water. And this is also due to this hydrogen bonding. So the unusual thing about water is when it gets colder, the molecules actually spread further apart. And so as they don't move as quickly, so their temperature goes down, that means the average speed of the molecules goes down, they kind of settle into a large lattice framework. And so that means their density is lower when it's in liquid water form. When the temperature is higher, the molecules are able to move closer together than they do at lower temperatures. And that's unusual. In other compounds that do not have hydrogen bonding, as temperature decreases, they become more dense. So this is a very unusual characteristic about water. When temperature goes down, they become less dense, and when temperature increases, they become more dense to a point. And this is very important for organisms that live in the water. So when temperatures fall, ice forms, and the ice layer, instead of sinking to the bottom of the pond, is going to float on the top. And when it floats on the top, what it will do is it will create an insulating effect. So the organisms that are below in the water, they will actually be insulated and they won't die during cold weather. If instead the ice was more dense when it got cold, then what would happen is ice would form on the bottom and it would slowly form ice all the way to the top, and all of that ice would have killed the living things. So it's actually a benefit, a great benefit to organisms that live in water, that when temperatures get cold, the ice floats on the top and it helps insulate and keep liquid water down below the ice for the living things. Another important characteristic of water is that it is a universal solvent. And a solvent is a substance that can dissolve other substances. And the important type of substance that water can dissolve are ionically bonded substances. So what water can do is water can dissolve ions. So remember, an ion like sodium or chloride is a charged atom. Because that atom has a charge, that means that water molecules will be attracted to it. So if you have an ion with a positive charge, then water molecules are going to be attracted to that ion, and they are going to be attracted with their negative, the oxygen side, oriented towards the cation. With the anion, a negative charged ion, the hydrogen part of the water molecules is going to be attracted to that ion. And so what basically happens is you are going to get a shell of water molecules surrounding the ions. And they call that a hydration shell. And being able to form those hydration shells, that's what allows water to dissolve ionic compounds. So things that are held together by ionic bonds, like salts. Sodium chloride is regular table salt. I told you that is a very strong interaction, and living things cannot break the ionic bonds holding the sodium and chloride together, and that is true. but because water molecules are so small, they can fit in the space between the ions. Remember, the ions are held fairly far apart, and they can form these hydration shells around the ions. And in doing so, that's going to pull the ions apart and dissolve the ionic substance. Another important characteristic of water, and due to hydrogen bonding, is called cohesion. And this is the ability of water molecules to hydrogen bond to each other. So this is what I've been showing you before. So these are all water molecules, and these are all hydrogen bonds between the water molecules. And again, you're going to have the hydrogen bonds between an oxygen part and a hydrogen part. Oxygen has the partial negative charge. Hydrogen has the partial positive charge. And so this is cohesion. So cohesion, the ability of water molecules to sort of stick to each other, to adhere to each other. And so this is why when you see drops of water falling, they fall in beads, because the water molecules aren't just going to disassociate and fall apart from each other. They're going to hold together with that hydrogen bonding. And that gives all sorts of characteristics, like you can almost overfill a glass if you filled a glass with water. So if this is the glass, you can almost overfill it with the water is kind of beading over. That's exaggerated, of course, but that's because you have that hydrogen bond holding those water molecules together. And also that creates surface tension, like you see this coin floating on the water. Here's the beading. You can see this in nature. So that is a stem releasing water. And again, the water molecules aren't just falling apart, but they're being held together by cohesion into a bead. And that allows the surface tension, some insects. So those insects just skate along the top of the water. So cohesion gives rise to the ability of surface tension, water to have a high level of surface tension. Related to cohesion is the idea of adhesion. And adhesion is the ability of water to hydrogen bond to other types of molecules. So these would be other polar molecules or charged molecules. And this is the idea of capillary action. So here you have some water with some blue food coloring in it. You take a paper towel and just touch it to a corner of the water. Because the paper towel has a charge to it, the water will adhere and be drawn up into the paper towel. So that's capillary action. It's due to the water molecules adhering to molecules other than water. And this is very common in plants. So plant stems have charges to them. So when the plant stem is put into water, the water is going to adhere and move into the flowers. So of course, if the water has food coloring in it, eventually as that water moves up into the plant, it's going to cause the petals to turn a different color. And this is an example of a capillary tube that it's so thin and glass has a charge to it that when it's placed in the water is drawn up the capillary tube. And that's actually how human capillaries work with blood flow in the body. Because the capillaries are so thin, the blood, the liquid part of the blood, the water in the blood is attracted to the sides of the capillary which have a charge, and then blood flows through the capillary. Cohesion and adhesion are actually very important for getting water to flow through plants. So especially trees, which can be hundreds of feet tall, it's very important for the leaves at the very top of the plant to get water that is drawn in from the roots and to travel all the way up against gravity to the very top of the plant. And to do this, you need a combination of cohesion and adhesion. So adhesion, the water molecules are attracted to the sides of the cells in the plant, and cohesion, the water molecules are attracted to each other. So it's kind of like a train. A train of water molecules is being attracted to the charged sides of the vessels in the plant. And this is an animation that kind of shows you. So again, cohesion, holding those water molecules together, adhesion, drawing that water, sort of that water train all the way up to the top of the plant. Another important aspect of water is that it has the ability to organize nonpolar molecules. So nonpolar molecules, these are molecules that do not have a net charge on one side or the other. And the example of this, I'm sure you have all seen, is that when you mix oil and water, they don't mix. So what happens is initially when you pour the oil into the water, they sort of mix, but you will see these beads forming. And that is because water will not mix with a nonpolar molecule like oil. And eventually, when this all settles, you're going to end up with a layer of oil sitting atop and separate from the water that is below. So that's organizing the nonpolar molecules. The water does not dissolve them, it organizes them, it separates them. And this will be very important when we talk about cells and the cell membrane, because the cell membrane is made of nonpolar molecules. So when you mix these phospholipids, these nonpolar molecules in water, they will spontaneously form a cell-like structure. So this is basically the structure of the membrane, where you have the water in the environment here and the water inside the cytosol that's inside the cell there. So that ability of water to organize nonpolar molecules actually gives rise to the basic cell structure. The last characteristic of water that I want to talk about is pH, and this occurs in liquid water. And in liquid water, some of the water molecules are going to spontaneously disassociate or break apart. And that's where a hydrogen disassociates from the other oxygen and hydrogen. And this is a very dynamic process, and it's reversible, so often it's drawn like that. And relatively speaking, it's very few molecules that actually disassociate this way. And in reality, what will happen is when that hydrogen breaks apart, it's actually going to join with another water molecule for a very brief period of time. So actually what you end up having is a hydronium ion forming and also your hydroxide ion. And this is very dynamic. This is happening all the time. It's breaking, reforming, breaking, reforming. Now pH is a measure of how much hydronium ion is present. So technically pH is the negative log of the concentration of hydronium ion. Now most of the time we don't refer to this as hydronium ion. Instead we just write it as the hydrogen ion. So basically pH is a measure, it's the inverse log of the concentration of hydrogen ions in liquid water. The pH scale is a measure of pH ranging from 0 to 14. And remember, pH is the inverse log of the concentration of hydrogen ions. So because it's the inverse log, that means the lower temperature, the higher the concentration of hydrogen ions. So if something has a pH of 1, that means it has a higher concentration of hydrogen ions compared to something that has a pH of 14, where you have fewer hydrogen ions in the solution. So 7 is considered neutral, and that's where the concentration of hydrogen ions is equal to the concentration of hydroxides. So there you have an even distribution of them. If you increase the amount of hydrogen ions, the pH is going to decrease, the pH number is going to decrease, and it becomes more acidic. If you reduce the concentration of hydrogen ions compared to the hydroxides, then it's considered a basic solution and the pH number is higher. Because again, remember, the pH number is the inverse log of the concentration of hydrogen ions. The other thing about the pH number is because it is a logarithmic scale, that means between numbers it is 10 times higher or lower depending if you're going increasing or decreasing. So battery acid has 10 times the amount of hydrogen ions, concentration of hydrogen ions compared to gastric juice or lemon juice, even though they're just one number apart. Again, because it's a logarithmic scale, the differences between numbers is tenfold. Most living things prefer a neutral pH, so you'll see that a lot of solutions that are related to living things, like seawater, human blood, tears, pure water, saliva, urine, they all range around neutral. Things that are too acidic, like battery acid, can be damaging to living things, as well as things that are too basic, like oven cleaner. There are some substances that have the ability to change the pH of a solution, and these substances are referred to as acids or bases. If a substance like HCl is added to a solution, it will increase the concentration of hydrogen ions in that solution. And in that case, it is considered an acid because it's going to increase the concentration of hydrogen ions, which will decrease the pH of the solution. Conversely, a substance like NaOH will decrease the concentration of hydrogen ions, the relative concentration, by increasing the concentration of OH in the solution. So this will increase the pH of the solution overall and making it more basic. Buffers are substances that maintain a constant pH. In other words, they resist changes to pH. So a buffer is a substance that can absorb or release hydrogen ions to maintain a constant pH level. Most living things require a neutral pH. So most living things have the ability to buffer the solution inside their cell and inside their bodies. For instance, for humans need to maintain tight control over their blood pH levels. A normal pH for human blood is about 7.35 to 7.45. If there's a change in that pH, if it starts decreasing, the person is starting to go through acidosis because the pH is decreasing, going more towards an acid. If the pH starts to increase, then it's alkalosis. And if the change is too much, it can lead to death on either way. So really, the normal range for functioning for human blood is from 7 to 7.8. If the pH changes, goes too low, below 7 or above 7.8, that will result in death. To maintain our blood pH, we have a buffer in our blood called bicarbonate. So bicarbonate will buffer our blood, so if acid enters, it's going to absorb the acid, or if the blood becomes too alkaline, it's going to release hydrogen ions. So that is one of the major buffers that maintains the pH of our blood so that our bodies can function.